Ermentrout to speak. It's an international talk. Bart is in Pittsburgh. I'm in California at the moment. Suresh is in Montreal. Michael Mackey is in Germany. So there's an international audience listening. Bart did his PhD at the University of Chicago, and he got there in 1975, I guess, which is a few years after I left there. He was doing early work with Jack Cowan on hallucinations. From there, he went to the NIH and he worked with John Rinzel for a few years, and has had quite a distinguished career in the area roughly known as computational neuroscience. He's done a lot of work with Nancy Kopel and is really one of the most highly cited people to be working in this field. He's a lot of fun to see in person, so it's too bad that we're all separated by big distances, but maybe you'll get an idea of his spirit and enthusiasm through the talk. So it's a pleasure to welcome Bart to talk to us today.
Thanks, Leon. Just a note for the listeners, if you have any questions, just put them in the chat and I'll pass them on to Bart during the talk. All yours, Bart. Go ahead.
All right, thanks. So this is part of a long ongoing project that started with something called a NSF Ideas Lab back in, I think it would have been 2015, because I had lost my sense of smell through a series of viral infections. And it was sort of ironic that I was at this big boot camp for trying to crack olfaction, and I couldn't smell a damn thing. I have gotten it back since then. So this has been going on for a while. It's been extended to a NeuroNext project, which I'll describe shortly. All right, let's see. Why isn't it? There. All right. So olfactory search, detection of chemical gradients is essential for obtaining nutrients and avoiding threats. Turbulent fluid dynamics are common in macroscopic environments, but animals have robust methods for extracting directional cues from this variable olfactory signal. And so the question I want to talk about today is what behavioral processes allow for successful navigation, what kinds of algorithms could be used, and so on. And there's a lot of work that's been done on that because of its importance in robotics. I was going to show, I had some robotics videos. Damn, I was going to put them in there. Well, maybe I'll see if I can dig them out and show them at the end. So this is a long-lasting collaboration with a bunch of experimentalists, starting with the work I did with Nathan Urban's group, in particular his student Annie Liu and his postdoc Andy Papali, tracking mice on an open table which are trained to locate an odor. Also doing some work with Kathy Nagel's lab, where she tracks walking fruit flies, and Justus Verhagen, who's at Yale, he works on mice in virtual environments and also robotics. So here's the experimental setup. So basically there's a table, and underneath there's infrared lighting, and there is this transparent plexiglass. And what you can do is it's completely in the dark, so the animal doesn't have any visual clues. And the IR-sensitive camera can detect the warmth from the mouse. Down there are some anemometer readings just to indicate that there really isn't too much wind. So I'm going to first show you a video. All right. So soon you'll see Annie's hand come down, and there's the mouse. All right. The spot is over here. All right. The mouse finds the spot, and there's a little piece of peanut on it. And now the mouse continues to just chill and eat the peanut and then wander around some more. Okay. And here's some examples of successes and failures. So a failure is if after 3 minutes, the mouse doesn't find the spot, and otherwise it's considered a success. And these are just some examples of trails. The color coding here is time. So dark blue is the longest time ago, and yellow is the most recent. Similarly, mice can be trained to follow trails. Okay, the right odor is associated with a reward, and the left is just a sham trail. Incidentally, these trails are made with a wintergreen scented crayon. So you can see the mouse does a pretty good job of following the trail and now wanders off. May get back on there. So here's a series of paths of the mouse, and you can see that it spends most of its time on the trail. So what do real odor landscapes look like? Okay, well, the John Cromaldi lab at UC Boulder uses something called planar laser-induced fluorescence to image plumes in air and water. So for example, in air, what he does is he mixes acetone with, I guess with,
very cool.
hello? Oh, he mixes acetone with helium so that it's neutrally buoyant, and then flows that into the air because acetone fluoresces, and in water they can just use fluorescent dyes. Okay, and what we do is use these as test beds for our different algorithms. Question in the chat? Oh, just an accident. Okay. All right, I'll tell you, I guess I should just ignore the chat. And Suresh will tell me when something is important in the chat, right?
Yes, that's a good idea.
All right. Okay. So this is more realistic than the noisy gradients that you'll see later on, and easier than trying to solve the full Navier-Stokes simulations. So this is the air setup that he uses. Okay, so there's an intake, and across here's the laser, and there's the camera. And this is just, it's 2D images. We don't do 3D, just 2D. So here is a 10 centimeter per second airflow bounded. So that means that the flow is close to the ground. And so you can see it's much more diffusive. Whereas here it's 20 centimeters per second, and it's farther away from the bottom of the floor. And so that it's much more plume-like. Okay, and these images were done by Aaron Connor, who was one of John's Cromaldi's PhD students. So this is an instantaneous shot of a water plume. As you can see, the water plumes are much more complicated. There's many eddies and filaments and little vortices, and there's the source, and the water is flowing in this direction. So any algorithm that we're going to test is going to have to deal with this sort of fluctuation and lack of it's not a simple gradient type of picture here. So we're going to use behaviorally inspired algorithms. I'm not going to show you this data, but we've gathered lots of data for what the probability of how often the mouse moves, what the velocity of the mouse is, how his head moves, the probability of his nose being in a given angle with the body, and so on. So these models are sort of based loosely on this data, but before we actually try and connect it to data, and I have some other I do have a couple of papers if anybody's interested where we really do compare to data. This is a broader picture, and I want to throw some math in here as well. So typically in these models, the agent our artificial mouse there moves at a constant speed v, though in some models this does vary, in a time-dependent heading. So there's the differential equations. dx dt is v cosine theta, and dy dt is v sine theta. So the crucial point here is, how do we update this heading? All right. And typically there's three forces that undergo that are used to update the heading. One is the deterministic one, and that's some sort of odor comparison rule that says to adjust theta towards what you think is the more likely area or more likely part due to the fact that the odor might be stronger. The second force is flow, and the fluid flow direction, for example, wind, provides hints as to which direction the source is and contributes to F flow. And finally, F noise can be regarded either as sensor noise, because all sensory systems are noisy, or actually as a strategy for exploring when there's no clear odor cue. And typically, it will be white noise with a possible odor-dependent magnitude. Okay, so I'm going to go into more detail about which each of these, about F flow and F comp shortly. Okay, so here's F flow. All right, and if there's the flow is always, in all these examples, in all the plumes and things, the flow is always coming from the left, somewhere in the center. Okay, so we know that we want to go towards the left, if that's because that's where the source is going to be. And if there's odor and there's flow, then we should, if we sense odor and we sense flow, then we should orient towards the flow. But if we don't sense any odor and we know flow, it's a good idea to go downstream, since in general the plume is broader there. Okay, as you saw here, the plume is much broader downstream. And even here, up here, there's very unlikely to get an odor unless you're really close by, whereas here, there's a much better chance. So that's a strategy to move upstream. And we typically have a function that looks like this as a function of odor concentration. So above this threshold, we'll move towards the left, and below the threshold, we'll move towards the right. So this is this function G. Okay, so the first thing I want to talk about is something called tropotaxis. Insects such as ants, fruit flies, bees, etc., and crustaceans such as lobsters use their antennae to localize odor. The nostrils or nares in dogs, rodents, moles, and humans are close, but can preserve some spatial information. For example, despite close proximity, this is an example of particle image velocimetry in humans. There are two separate streams of odor going into the nose. This is somebody breathing in, okay? And you can see that they're well separated, so that in some sense you can detect odor almost like you do with your ears, detecting sound localization. And there's some really cool experiments done with humans where they ask humans to follow a trail, and they've got this kind of mask on with extended nares or nostrils. Okay, so this is an example of a person following the trail. Here's an example of a mole that is trying to find a worm. The worm is in this black spot here, and this is the normal path that the mole does. If you block the left nostril or the right nostril, you can see that it's much more difficult to get, and it doesn't take a direct path. Nathan Urban and his PhD student, Pete Jones, also showed that blocking the nostrils of a mouse causes the mouse to be either to the left or to the right of where the trail actually is. Another strategy is called clinotaxis, and that's similar to casting. And what you're doing in that case is you're sampling serially in time. Dogs, mice, and many other animals move their heads and bodies to better find the location of the odorant. Moths and other insects move orthogonally in wider and wider arcs to catch a plume. So that's kind of an example of casting and surging. So before we get into actually doing anything, some of the algorithms, we're going to be looking at local concentrations of the odorant, either in static odor landscapes or in image plumes. Recent work in our group, this is with Jonathan Victor and other people, has shown that the transformation of odor concentration can improve the information that a given sampling provides about the location of the odor. Okay, so there's a couple of papers if you want to look at it. And it turns out olfactory receptors from flies and mice transform concentrations before they even get to the brain. And many receptors have at minimum simple Michaelis-Menten-Hill type function responses, where the response is the ratio of the actual concentration divided by the concentration plus a minimum. So at low concentrations, this greatly amplifies the contrast. And of course, it saturates at high concentrations. Just 1 second, I want to get a little water here. There we go. So, what sort of algorithms could we use? Well, we're going to focus mainly on local algorithms that depend only on the current state and possibly a little bit of history. And the two that I'm mostly going to focus on are tropotaxis, where you've got two sensors and you simultaneously compare. So this is a spatial model. For example, using the differences in the two nares, nostrils, that is, or localizing sound with ears. And then the other one I'm going to use is clinotaxis, where you make alternate lateral movements of part of the body and compare successive intensities. And I'll often call this casting, even though it's technically not the same. Another algorithm that we've done some work on, but I'm not going to talk about today, is intermittency, where when you've got these very chaotic plumes, the time between hits of odor filaments gets shorter as the source is approached. So you can move towards where the time between hits is shortening. Finally, there are some global algorithms like infotaxis and entropotaxis that require a lot of complex calculation of the probability and a complete memory of where you've already looked. And in fact, for example, if I smell something and I don't sense an odor, then I can block that area off and say, okay, the odor is not there. So that requires kind of a global memory. And it's difficult to imagine how it's implemented biologically. So here's the first example, and this is the planar binaural algorithm. All right, there's the full set of differential equations. dx dt is v cosine theta, dy dt is v sine theta, and d theta dt is just the concentration between the left sensor minus the concentration of the right sensor. So if the left is stronger, you move counterclockwise, and if the left is weaker, you move clockwise. And there's a sensor length, and there's a sensor angle, and there's position of the body. So this works fine on smooth Gaussian landscapes. So for example, here's an example of a spot, all right? And these are trajectories of the agent, and you can see that all these trajectories, it gets to the spot. Here's one where it misses. Here's somewhere it misses. Can't compensate. Here's another example. The trail is along the y-axis, and you can see that it picks up the trail very nicely, In the purely deterministic case, if I'm going orthogonal to the trail, then the left sensor and the right sensor sense exactly the same thing. So there's no change, and I just cross right through. So we can actually do a little mathematics. When you're on a trail, all right, there's no y dependence because the trail is the y-axis, and this reduces to a planar dynamical system that is just in x along the distance from the trail and theta the heading. And what you can see here is that there's a single fixed point. There's actually two fixed points, one at pi over 2 and one at minus pi over 2. One is heading up the trail and one is heading down the trail. So I'll just focus on heading up the trail. All right. And that is a stable, spiral in this particular case. And interestingly enough, there's these blue basins of attraction. So anything that starts in here will end in here, and anything that starts out of this basin will not. And we were kind of surprised about this because usually when you have basins of attraction, that implies there's a saddle point. But here, there's two stable fixed points, pi over 2 and minus pi over 2, and no saddles. So how do you explain where this basin comes from? Well, you can do a little expansion. For small sensor length, this becomes a simple differential equation, again, much simpler than what I saw. And you can quickly see, if you look at the ratio of x over dx dt over d theta dt, you can see it factors. And so this is what's called an integrable dynamical system. And you can write down a kind of energy function. And the level sets of that essentially determine the basins of attraction. So the level set that corresponds to pi over 2, theta equals pi over 2, E equals 0 is this red curve, and that's really close to the basin. So that's sort of our explanation for the basin. What about spot finding? Well, a radially symmetric landscape is very simple because we can then convert to polar coordinates. And then instead of looking, we still have a three-dimensional system, r, phi, and theta, but now we look at the another variable psi, which is a function of theta minus phi. Theta is your heading and phi is your polar angle. And again, this reduces to a two-dimensional dynamical system for which there's two equilibrium points that correspond to periodic orbits around the spot. Neither are stable, but there's a localized region near the spot that's attracting. So as far as the mouse is concerned, all it has to do is get close. And here's an example of what I mean. Okay, there's this unstable limit cycle here. The odor is actually at r equals 0. Okay, and basically, if we start, you can see that we can get arbitrarily close to the spot. Okay, just by following any of these black trajectories. There is this unstable torus here, which is kind of cool, that keeps you out of this unstable limit cycle. And basically these stable manifolds, these blue trajectories of the saddle point or the saddle limit cycle here, determine whether you'll make it into the spot or exit the spot. So how do these algorithms do in real plumes? I'll play these again. Okay, the left is that broad plume, and this is the binaural, and you can see it gets very close. Let me try it again. All right. All right, and you can see they do pretty well. Any questions yet?
None, it seems.
Okay, so this is a water plume. And it's much more turbulent, all right? So in this case, we've added an additional force, and that was the pure binaural, the one you saw with air. Here, we've added an additional force, which is some clues about the direction of the flow, okay? So that's what you're seeing there, because otherwise what will happen is if it finds a filament, it will chase it backwards. You see an example of that right here, okay? There's the agent, and there it's chased a filament backwards a little bit, but then because of the airflow information, it's compensated and turned around. And even though these concentrations are intermittent and very faint, this algorithm does a pretty good job of actually reaching the target. So I want to turn now to search strategies, okay? Far from the source, there's no odor. So what sorts of strategies could be used? There's various structured walks like spiraling outward. You can do correlated random walks, which is the same thing as we had before, but the heading also has some white noise to it, and also things like levee flights. Humans themselves use many strategies. So this is a really cool experiment on humans done by former collaborators of ours, Lucia Jacobs and her students, her student Judy Jin. No, that's Judy Jin. Typo there. Okay, what they did was we have the actual plumes that John Cromaldi imaged. All right, and what they do is they have headphones on, and so that when they get close to when they sense the plume, then when they're in the plume and they're left, they get information about that by clicking in the headphones. So it's sort of like you're getting warmer. So here's some human strategies. Can you guys see these? They're pretty faint. Can you see the little gray lines?
I can see them.
All right, good. So here's one where a person just zigzags back and forth. Okay, here's one where this is the starting location, and that red circle is the plume. Here's one where the guy goes back and forth like this until he catches a little bit of it and then zooms in. Here's one where they do the edge strategy. Here's one where it just goes back and forth. This one was kind of completely random. Here's a kind of spiraling outward. So these are all different strategies that human subjects make. Here's an example of a mouse trajectory. All right. Here the mouse starts here, wanders around. This is missing data and then gets to the endpoint. Okay, here's an example of simulation of what I'll call a Roomba search. Just spiral outward until you get some hint of the odor, and then you go find it. So this guy spiraled outward until it until it sensed it somewhere around -40, plus or minus 40 40 time space units away. And then it gloms in.
Ask a couple of questions, very naive
Sure.
questions. So first, I guess with this pattern, like why are there these spiral patterns even once you come close to the plume, given that all the plume...
That is because in the binaural odor search, it's not a plume, it's a single point source, and the velocity is never zero, so it doesn't stop. So it just zips around. This is there's a torus bifurcation here, and it just undergoes a torus.
And in the previous one, it deviates. It seems to be going straight towards the source, and then it deviates, does this whole, in the immediately previous figure that you had. You're way behind.
Okay, I think I see what you mean. Yeah, I'm going to see if I can get to it. There, like that, right?
No, the one before the one we were looking at.
Oh, the one here?
The example that you had. No, if you keep going towards the one we were just looking at. I'm lost now. No, I think it's far ahead. You went way behind.
Uh-oh, uh-oh. This is the trouble with these videos. It really screws me up here.
Yeah. Sorry about this, but
No, that's okay. I've never had this problem before on Linux, and something about, I never had this problem before on the Mac either. Now I hope I can get out of this because I can't even kill this process. Crap. All right.
Ah.
There's the Roomba search, but
And
I have
was
to
the
get
one-
rid of these. Let me
Yeah.
see if I can get out of here somehow.
sorry.
Now it's these videos. Here, it looks like I might be. Nope, I'm still. All right, I'm gonna start over again here.
Yeah.
Oh, wait, maybe I'm good. I'm good now.
Yeah.
All right. Am I sharing still?
Yes, you are.
Okay, so let's go back to full screen mode. All right, so let me go back to what you're asking. Try and avoid the videos. Come on. I don't know what, I'm gonna have to kill this thing.
I should have waited till the end.
No. This. All right, force quit. Never used to have this problem until I got this new Mac. This is supposed to be much faster. All right, let's try again. Let me find it. There we go. All right. The Roomba search, is that where you want it?
Yeah, exactly. And actually the slide before the Roomba.
Okay. That one. That?
We don't see it though right now. You have to screen
I'm
share.
not sharing anymore. All right, let's see. Where's the share screen? Where the heck's the share screen button? Here. Sorry about that. This is a. Maybe I should have just stuck with Linux. I don't have this problem. All right. Okay, so
This
That-
one, exactly. The one immediately before.
that's this one.
Exactly, yes.
Okay, that's a mouse. That's a real mouse.
Oh, yeah.
Okay, that's not one of my strategies.
Got it.
Yeah, mouse trajectory. Yeah. Okay. All right. If I had known it was only one back, I wouldn't have screwed up. Okay. All right. So there's Levy's flight. All right. And that's another example. You guys know, I mean, that's just you do a bunch of short turns and then you make these long runs. So noise helps with real plumes too. You can see on the left is with no noise, and it's just going to go back and forth like that. Whereas on the right, it successfully finds the plume here. I think I can do that again. And it does a great job, and there it goes. Now I'm going to stop that. There. All right. So I don't know why this slide looks like it's only got half the stuff. All right. So that slide seems like in the, uh-oh. What? Sorry, I really shouldn't go. Don't let me go backwards again. This is really annoying. This does not. All right, I guess I should use PowerPoint, right?
Yeah.
All right. There, it's finally finding it. Maybe. Oh, come on. I'm sorry. This is,
I think we just need to get out again and come back. Yeah.
yeah. This only happened on this new Mac. No, I don't want to quit Zoom. All right. One more time. This time, no more.
No more reversing.
No more damn videos. All right, let's try one more time. It's weird because all right, now, option L. Before I do anything, let's get down to here. Okay, noise helps. All right.
Share screen. Share screen.
I'm going to do that now. I can find the share screen button. There.
Yeah, works.
And then L. All right. Hopefully there's no more videos. Okay.
Excellent.
All right. So far enough from a spot, psi equals 0 and pi are attracting, so the agent just goes off to infinity. The basin is similarly small for a trail. So one search strategy is just a correlated random walk. That is noise in the heading. Okay. So here's an example, a rather obvious example. Here I started outside the basin of attraction. There's the basin of attraction, the blue curves, and the noise drives me back in, and then I find the trail. Similarly, find the trail, okay? And this is in the XY coordinates. Similarly, with a point source, noise, which here normally I would go off to infinity, but here the noise drives me in this way. Another example, I'd be going off to infinity, noise drives me towards the source. And this is what it looks like in XY. So what I want to do now is in each model, 50,000 points are sampled in X, Y, and theta and run for a fixed number of iterations or until a boundary is crossed. For spots, the domain's the annulus. One is R min less than equal to R less than equal to R max. And for trails, minus L to L. Domain is binned and colorized according to the probability of success. And the probability of success is summed up, and the mean time to reach the target is averaged over that given when you succeed. In most cases, we average the success as a function of the noise strength, okay? So this is a great example of stochastic resonance, okay? So here is a constant noise with no hill, all right? So this is very low noise, and you can see that basin of attraction. This is a spot. And now as I add a little bit more noise, some of this fills in, and you can see the probability of success goes up a little bit, all right? And whereas the time to get there always increases because the noise makes the trail more tortuous, okay? When we add a hill, what happened? Did that just crash? Jesus. I'm really sorry about this.
It's all right.
I've been giving talks over Zoom now for 3 years, and I've never had anything like this happen. Sorry, an error. Well, I guess you get what you pay for. It wasn't even a movie. All right. All right. Am I still sharing?
Yes, you are.
All right. I'm almost done, thankfully. All right. So with Hill function, I get better success, and there's still a resonant noise. Okay. Similarly, if I change the parameters of the Hill function, you can see I get monotonic increasing in success up to 80%, as opposed to with no noise where I can't get above 60%. What about real plumes? And I thought this is a really great example of resonance. Here, what I do is I start with I think 1,000 points randomly distributed in this plume. And now what I do is it's a failure if I go off any of the edges, and I got to get within a few pixels of the source. All right. And without any noise, I have 20% success. But then as the noise level goes up to 1, I get up to 50%. What really I liked about this, as opposed to the static Gaussian plumes or Gaussian landscapes, this is by far the sharpest example of resonance. I don't know whether there's some time scale stuff going on here, and we have to do this with some other plumes to see if there's some sort of relevant scaling here. So other questions you can ask is, can we improve on this? Once we're in the basin of attraction, maybe we should shut off the noise, and outside the basin, then the behavior should be more noisy. But we can't know the basin, so we use the mean concentration of the sensors as a surrogate, and instead have the noise decrease when we're close to the source. And here you can see that there's this nice monotonic increase on the probability of success, as well as a decrease in the time to success as we increase the sensitivity to the noise, to the concentration. Similarly there as well. And similar results on an infinite trail. So basically, and with a Hill function, I have no idea what's going on. I ran through this talk 3 times today.
I'm sure. I mean, it's only a few more slides, so I guess we just keep doing this.
It is. You know what? I'm not even going to go to full screen. Okay. All right. Well, I don't know what to say. I'm going to use Linux from now on and Ocular, which never crashes. All right. So I'm sorry, this is really frustrating. But basically, noise in real plumes can extend the basin without affecting the time to get there. So here's the basin with no noise in a real plume, and we're increasing so each dot here indicates a success, and the color is how long it took to get there. So blue took the least amount of time, yellow took the most amount of time, and red took even longer. But you can see that by the time we set sigma equal to 0.6, we can start anywhere in here and get almost 100% success. So noise extends the basin without affecting the time too much. I'll skip the clinotaxis of the casting. I will just tell you at the end, nothing works. Nothing works better than binaural. All the casting, all the other options, binaural works better than anything in every case. All right, conclusions. I really apologize for this. A simple tropotactic rule is quite successful in model and real landscapes. Certain amount of noise can improve the probability of success. Smart noise, such as concentration dependent, leads to greater improvement. And noise can improve clinotaxis and casting rules as well. Local algorithms are really fast, easily implemented with simple neural circuits. And let me close with some acknowledgements. And I do want to show you a robot video, okay? Just as a last ditch thing, if I can find it, and that doesn't involve Acrobat. Okay, so let me give you the acknowledgements. All the mouse work was Nathan Urban's lab, and James Hungenius was a postdoc of mine. Noor Riemann was a grad student. Casey O'Brien was an undergrad. Boei Ouyang has done all the water work. The John Cromaldi lab, Aaron Connors and Aaron True. Jonathan Victor and Sebastian Boy have done did the information theory stuff. Kathy Nagel, I didn't show much of anything of her stuff at NYU, and then NSF Ideas Lab and NeuroNext. So I will stop this here and let me see. I think I can find, I'm hoping I can find some, let me see if I can find a robot. Ah. All right. So this is, I'm going to see if I can get this bigger. I don't know if I can. All right. So here's the setup. There's a robot. All right. You see our little robot? Is my screen showing?
Yes.
All right. And this is controlled with some PIDs. These are ion detectors. Okay. And it's got one on either side, and there's a little Arduino controller on here. All right. And there's 3 possible locations for an odor source. Okay. And so we're going to see if our robot can find it. And it's based on, it's using this kind of binaural algorithm. That was a failure. Up there. There we go. Success. Another success. And I think this will be this might be a failure. Yeah, yeah. Yep. And one more. Yeah. So there we go. So basically, I'll stop share here. We can actually put these algorithms into real robots, and they're autonomous and they can actually find odors. So that's another advantage of this. The nice thing about test beds is that about testing things on these odors on the test beds is that we can check the algorithms, we can refine them and before plugging them in robots. So again, I really want to apologize for all the technical difficulties. This has never happened before. Maybe it's a Canadian thing. I don't know.
All good. It was a great talk, and we are well in time for questions. So we have time for questions. Anyone on the chat, or you can
just don't make me show anything with Acrobat.
Yeah. While someone is looking for a question, I'll ask one. So I was actually trying to understand how well the strategies and which of the model and also of the mouse have knowledge of the sort of geometries they're likely to find in the environment. So one thing that struck me is I don't know the physics, I'm ignorant of the physics, but the plumes always seem to be going in completely linear, like a very, very tightly bounded along a line, right? Even if there is a source. And I was wondering if the strategies are making use of this knowledge because are they aware of this?
Okay, in fact, okay, here, our strategies, the only knowledge we use is the flow direction, okay? And that's only for the water plumes. For the air plumes, we don't even use that. We pretend there's no knowledge. Now, it's interesting that you asked that, because I mentioned this algorithm called infotaxis, okay? Infotaxis has built into it, it has to have a prior about what it thinks the shape of the plume is, okay? And of course, if it's wrong about that, then the algorithm fails really miserably. So it has to have a good model for the plume, okay? So then it does know it. My student, Bo Wei, who was here, is giving a talk Thursday in our math bio thing. And I believe he's going to show some examples. We have some examples from John of the water plumes where the source is moving up and down, all right? So then the plume is much more complicated and it's a constantly moving source. And there, knowing what the plume looks like would probably, or having a prior about what the plume looks like would probably fail miserably. So here we actually are able to follow this moving source pretty well, I think.
Is there any evidence that mice know about this, like real mice?
I don't know if they know what I don't know how you would know. There are some ingenious experiments that other groups have done where they put the mouse on a treadmill, okay? And that way they can vary this odor trajectory all over and watch how the mouse follows it. And there it's just on a trail, there's no plume. So in that case, you don't really need a knowledge of it. You just got to keep your nose to the grindstone, as they say. But I don't know if there's knowledge that they know what the plumes look like.
Or they learn it, right? Once they're in an environment.
Certainly you can say that in a sense, flies do, because Kathy Nagel's work has shown that the flies, when they sense wind, but no odor, then they move downwind, okay? And they actually know the neurons that the cool thing about flies is you can know every single, they know what every neuron does, okay? They don't know that in mice. We don't know whether there's evidence that mice pay attention even to airflow, okay? Whereas the flies, it's extremely important because when you're a little tiny fly, airflow really matters. So then what happens is, so there they have a knowledge, at least that they know if they go downwind, they're more likely to encounter the plume.
Great. Benjamin, you have a question. Would you like to ask it yourself? Or should I read it out?
Yeah.
Yes, so I can say, sorry, just needed to jump to the microphone. So yes, firstly, thank you very much for your talk. And I just had a question on the binaural algorithm. So I think you might have alluded to this at the end with the video and the robot, but did you consider a model where you had a PID controller? So in addition to just considering the difference between the two nostrils, just adding in the integral term and the derivative term? And if so, did you simulate this?
I'm sorry, I don't quite understand what you mean. So I have a PID, two PIDs?
So I was thinking a PID between each nostril. So like having
Yeah, PID on each nostril. Okay. Is that what you mean?
No, so I mean actually between the two nostrils, because you have the difference in
Oh, I see what you're saying. Yeah, actually, one of our big group has actually developed, Lisa Stowers has a head-mounted PID that she can put on a mouse.
Wow.
That they can now I will say that the PIDs are far less sensitive than the mouse is. Okay. but there's a hope that we can sort of see figure out what exactly the mouse is sensing. These are like little tiny head-mounted PIDs. So that's about as close to what I think to what you're asking we have. The case of actually using the algorithms and the PIDs, that's exactly what was done in the robot. But the mouse is always that the paper that involved the robots also did some experiments with mice, and the mice are a hell of a lot more accurate than are the robots. The robots are, you know, the mice do much better.
Okay, thank you very much.
Questions? So while we're waiting for another question, I would like to go back to I was wondering if, did you ever show data comparing your model to the mouse behavior? Like how well does the model fit the
okay.
mouse?
I didn't, but we have a paper that does that. If you were interested, if you look for a paper with LIU as the first author and me in there in Frontiers, okay? We quantified all kinds of aspects of the mouse, and the model's a little more, the model combines both casting and binaural. So it actually uses casting, but the casting is biased by the binaural stuff. So it's it's a more complicated algorithm than what I talked about here. But yeah, and there we did. And even more to that is the work in Kathy Nagel's lab, where because they have recordings from the neurons and they know what each neuron does, the trajectories that they generate with their model really matches the trajectory. I mean, you know, to the eye. I mean, I don't know if you can quantify it. We've quantified it with a mouse only in very crude terms, like tortuosity of the trail, probability of turning, velocity, and things like that.
Yeah, and I would go back to my other question, which is, again, I'm very curious about. So you were wondering how it can be done. I was thinking about how much the mouse can learn the properties of a given environment. So once you're in an environment, the plumes have, you're in water, right? It goes in a line. You're in air, it goes in a different way. Do mouse show different prior strategy, show that they're adopting different priors depending on the environment they are in?
I don't know.
Yeah.
I don't know the answer to that. When it's spot finding versus trail finding, we really hadn't done a whole lot of work on, we mostly, there was work with Pete Jones and Nathan did years ago, but then Pete dropped out of grad school and just stuck with went to med school.
Yeah.
And so that work was never finished. We do have some, I didn't show you this, but we've got some work on Y trails, okay? Which is kind of, you can think of as a different environment where we are trying to quantify what the strategy it does when the mouse comes to a Y, okay? Which way does it go? All right, and we can compare that to the models as well. And my former student, Noor Riemann, was analyzing a whole lot of that data. Again, the problem is Nathan's lab here, he kind of disappeared off the face of the earth.
Yeah. So Leon, Leon has a question or comment.
Leon.
So thanks, Bud. That was really fascinating. In your models that included noise and where you get to the source eventually, reminded me of Gambler's Ruin problems, where how long are you going to go on a random walk until you run out of money? And I just wondered if you ever tried to do anything analytical about computing the probability or the times to reach the source in some kind of biased Gambler's Ruin type analysis.
That's an interesting question. At first, I thought you were going to how does it compare if I just randomly walk around and allow myself to bounce off of the sides? And it does, you know, you get like 5% success rate in that case. So it does a lot better than that. We have actually posed this as a first passage problem.
Right, right.
Okay. And you end up getting a linear PDE, as you might guess. Okay. And we've actually solved it using something called Phoenix, which is a finite element PDE solver. Unfortunately, it takes so much longer to get it to work with Phoenix because it's Python and stuff than doing the Monte Carlo in MATLAB. That we actually abandoned doing the Phoenix in favor of the Monte Carlo. Analytically, I don't, maybe I should talk to you. We have not been able to figure out anything analytically yet.
You know, I think that Feller's book probably has a chapter on Gambler's Ruin. I haven't looked at that for a long
Look,
time.
Feller, you said?
I think so.
I have that book right over there.
Anyway, thanks a lot.
That's great. And thank you very much, Bart, for that fantastic
Yeah, and
talk.
again,
Really
sorry for all
interesting.
the technical-
No, it was not an issue. It added some flavor, kind of thing. And thanks everybody for coming and looking forward to the next talk. So great. Thanks everybody. Bye.
Okay, bye.